Hello, everyone. Thank you for joining us today. I'm Rachel Wong. Wan-Qing Yu and I will be discussing with you our use of semi-automated serial EM methods to reconstruct cells and circuitry in the retina in order for us to study the development, function, and repair. Now, I think most of you know that mature circuits in the nervous system form very stereotypic patterns of connections in the adult. The generation of these stereotypic patterns of connections actually arise during development, often by the elimination of connections with some cells while increasing connectivity with other cells. We also know that in injury or in disease, neurons die, and often this leads to eventually rewiring of the surviving neurons. These rewired or remodeled circuits pose a challenge in our attempts to repair or rebuild circuits upon reintroduction or transplantation of new cells to form or try to reform circuits that have been lost. Our lab's been really interested in cell-cell interactions and mechanisms that lead to the generation of these stereotypic wiring patterns by maturity. And in recent years, we've also been interested in the process of rewiring as a first step to try and understand what might be the constraints in rebuilding circuits during repair. Now, the circuits we study lie in the neuroretina of vertebrates. The retina is this light-sensitive part of the eye. And you can see that the retina is very, very well structured, and that it forms many layers of cells that are interconnected. Studying the retina is also important not only for understanding vision, but that in diseases of the retina, just like in other parts of the brain, cells remodel in their structure and also in their connectivity, giving us some insight into the neurodegeneration that occurs in the brain. The vertebrate retina, as I mentioned, is really arrayed in these very nice layers of cells. There are 5 major classes of neurons, photoreceptors which capture the light that passes on information to the bipolar interneurons that then excite these retinal ganglion cells that send their connections to the brain. Information flow from photoreceptors to bipolars to ganglion cells are modulated by horizontal cells or amacrine cells, interneurons in the outer and in the inner retina. Now, although there are only 5 major classes of retinal neurons, if you take any one class, you'll find that there are many, many types that comprise that class. For example, there are so many types of ganglion cells, and this therefore brings up this challenge for those of us in the field to try and not only identify all the types, but also their circuitry in order for us to truly understand how collectively these output neurons, for example, collect information about the image and pass it on to the brain. How do we map circuits that might underlie specific functions? Well, there are many ways, but here are just 2 examples. One showing here is using whole cell electrical recording techniques where you have an electrode that's patched on the cell body of a neuron. And then in this particular case, you can either target the cell because it's been labeled in a transgenic line, in which the cell expresses green fluorescent protein, for example, or you can identify the cell later by filling the cell with dye during the recording. You can then present light stimuli and look at the electrical changes or the voltage changes in response to those stimuli. Another method is to use calcium imaging. And here again, you can use calcium dyes or you can use genetically encoded calcium indicators. And what will happen is that these indicators, one way or another, will show changes in fluorescence over time. And these changes in fluorescence are really marked changes in intracellular calcium that are surrogate for neural activity. What about mapping circuits? Well, there are again many ways to do that. And we use a method where we can deliver plasmid using a gene gun, place the retin here, shoot it very gently, and cause individual cells here to express fluorescent protein either to fill the whole neuron, so you can see its arbor, and at the same time, have the cells express synaptic proteins, in this case, a postsynaptic protein that is tagged with a fluorescent protein, either GFP or another color. We can label these neurons, this is a retinal ganglion cell, in the background of transgenic lines where their presynaptic cells, the bipolar cells in this case, are colored because in a different color by expression of a different color fluorescent protein. Such fluorescent microscopy used for the reconstruction of cells only give us resolution in the micrometers or sub-micrometer range. So while we think that each one of these dots might represent a synapse, we really only know if we use methods that give us nanometer resolution in such a way that we even see synaptic vesicles. And one such method, of course, is electron microscopy. There are now numerous studies using electron microscopy to correlate structure with function. And you can imagine that if you just use EM to get a single image of a plane, this is not sufficient. In order to reconstruct the entire cell, or at least its connections, we have to do serial EM to obtain a volume reconstructions beyond just individual synapses like this. There has been, over the last several years, an increase in the number of techniques that enable us to do semi-automated larger volume reconstruction of tissue. One is using transmission electron microscopy, in which there is an array of cameras that are now located beneath the microscope so that you can image many, many sections at the same time. There are also methods that use scanning electron microscopes, such as those shown here. And one such method is the atom method, where serial sections are cut and they are placed onto or collected onto a film. And many of these are then arrayed onto a wafer, put into the microscope, and then scanned. There's yet another method called serial block phase scanning electron microscopy, in which you actually have the microtome, the bit that actually does all the cutting, inside the microscope, inside the scanning microscope. How this works is that the tissue here is being cut by a diamond knife, and as the section is removed, the block face is then imaged by the microscope, and over time you get a collection of all these images that stack up and produce a volume. And Wan-Qing will be telling you a lot more about this method, which we use quite a bit later on. Now we've used scanning electron microscopy methods, the serial block phase methods especially, for many discoveries. The first of which is to identify new cell types. And there have been numerous studies by various groups, the inventors of the block phase microscopy methods, Winfried Tang's group, to identify different interneurons, and also other groups, Sebastian Sung's, for example, this IY museum, adding to the collection of retinal ganglion cells from other techniques. We ourselves have used serial block phase EM microscopes together with fluorescent imaging to discover new cell types. For example, this cell is called GLUMI, glutamatergic monopolar interneuron. It actually looks like an amacrine cell, right? It's kind of bald here and has an arbor here. But we weren't really sure because it shared some molecular profile, as these are the neurons, the bipolar cells, which are connected to photoreceptors. We had a line where some of these cells express fluorescent protein. And then what we did was we used a method to mark these cells, and Wan-Qing will tell you more about this, locate the cell under EM, reconstruct the cell, and we actually found that these cells, the GLUMI cells, bear hallmarks of synaptic markers that often are found in the bipolar cells. So they have these ribbon structures at synapses, which are found in bipolar cells, but these neurons actually also have them, which is unusual and not what, for example, amacrine cells have. Other groups, for example, Kevin Brickman's group, have used combinations of calcium imaging to first of all record the responses of many ganglion cells here. And these responses are shown here. These different cells actually respond very well to direction movement of a bar in different directions. So in this direction or in this direction. And there are 4 major directions represented. They then basically fix the tissue for serial EM, reconstruct the cells from which these responses have been recorded, and then find the circuitry that actually underlies the cells connected to these ganglion cells that give rise to these various responses. We've used zero block phase EM to also look at circuit development, in this case, the human retina, especially in this region of the fovea. This is the region of our eye that we use to read the fine print of newspapers. It's high resolution, high acuity area. And normally in this region, a single photoreceptor cone is connected to two bipolar cells. Each are connected to a single ganglion cell, so that each of these ganglion cells actually have the resolution limit of an individual photoreceptor. This is the so-called private line. In development, however, the bipolar cell is reaching out to more than one cone, so it's not very precise. And each axon actually connects to more than one retinal ganglion cell. So here's an example where there has to be elimination of connections during development in order to form the precise or stereotypic pattern of connection at maturity. We've also used combination of fluorescent imaging and EM to look at the role of neurotransmission in our studies of synapse development. So for example, here's the cell body of a ganglion cell. And we can see some receptors here, inhibitory receptors that attack to fluorescent proteins in these cells. Normally, they're very small clusters, but in a mutant where there's no inhibitory transmission, these clusters are very large. However, we did not know whether these were synapses. So we had to basically take these cells, have an image of their clusters, mark the cell, do the zero EM reconstruction, and look at whether in fact, there are processes that are opposed to these receptor clusters, and indeed we find so. They are likely synapses. One can then also take one of these basically processes that connect to this cell and reconstruct, go all the way back and find the morphology of the individual cells that connect to this cell. So here's another example of using fluorescent imaging by tracing connections from individual synapses now back to, in fact, the cell types that connect at the synapses. Finally, we've also been using serial EM methods to look at circuit remodeling and repair. Here is an ongoing study where we've been studying the rewiring of, again, that private line, in this case, an injury in a non-human primate model. What has happened here is that we use a laser to burn off some photoreceptors. So only some photoreceptors and the terminals shown here remain. In another region, there are no terminals. We find that in regions far away from the lesion site, that there are a really strong connection between the bipolar here and the cone, and these are synaptic ribbons. But very close to the lesion, there's already remodeling. And here we can see that the cone looks a little different, and indeed is connected to more than one bipolar cell, so there is miswiring. Finally, we also been studying the reassembly of circuits. In this example, glial cells in the neuron have been encouraged to produce, glial cells have been encouraged to produce neurons in the mouse retina, and we were not sure if they were making actual connections. So again, using fluorescent microscopy together with EM, we can look at these connections, and even after the cells have been recorded. So I will stop here, and I hope that has given you a flavor of what this lab has been doing in using these techniques to answer specific questions. And now I'll hand it over to Wan-Qing, who will walk you through step by step, essentially how we combine these techniques to answer questions in the lab.
Thank you, Rachel. Now, I will briefly introduce our current analysis pipeline using a project I'm working on recently in collaboration with Greg Schwartz lab at Northwestern University. One of the goals in this project is to determine whether the two neuronal cell types receive similar or distinct connections from one type of presynaptic cell. We combined SEM with Leica microscopy largely because using LM, you can pre-identify and target cells of known structure and function for reconstruction. For example, in this image, two adjacent retinal ganglion cells with overlapping dendritic arbors were intracellularly diffused with Alexa480A, which is shown in green here, in a transgenic mouse line in which one type of the presynaptic bipolar cells express the red fluorescent protein TD tomato. The light responses of these two ganglion cells were also recorded. In order to relocate diffused cells in the EM volume, fiducial marks were burned into the tissue using near-infrared branding technique, essentially by line scanning at high power using a 2-photon laser. The retina was then processed for serial block phase SEM. We're mainly using serial block phase scanning electron microscopy, which was developed by Dank and Hans Holzman in 2004. This is the video clip from Zeiss microscopy demonstrating how it works. A microtome is mounted inside the microscope vacuum chamber. The sample is positioned on the specimen holder. A diamond knife removes a thin layer of the block face, and after each section, the exposed block face is imaged. The sample is then moved up in a small step. Sectioning and imaging is repeated in this way. And we can acquire images of hundreds of thousands, and then subsequently reconstruct it into a 3D volume. In this project, we actually sectioned and imaged the tissue volume of about 150 by 160 by 70 micrometer at the resolution of 5 nanometer per pixel, and each section is cut at 50 nanometer thick. We acquired 5 by 4 tiles with 10% overlap to cover the whole region of interest. Each tile is 8,000 by 8,000 pixels, which is about 40 by 40 micrometer. We cut about 1,300 sections, which took 6 weeks. The first step of our image analysis pipeline is imaging pre-processing using ImageJ, which includes converting the file format, bit depths, and also adjusting the contrast. We then create the 3D volume from the individual 2D images. We use TrackEM2 plugins in ImageJ to stitch and align all the image tiles. This is just an example of a single section of the stitched images. And usually using rigid transformation, TrackEM2 can perform fairly well. However, occasionally there is misalignment at overlapping region. This can be improved by further fine-tuning the parameters of the stitching program, and also sometimes iterations of the stitching might also be needed. Once the images are aligned, we reconstruct the circuitry mainly manually at present. This includes the 3D neuron skeletons and boundary segmentation. Here is a video clip showing how we manually reconstruct a neuron in TrackEM2. We first create the skeleton of the neuron of interest, and we need to make sure that we follow all the branches of the neuron. And then we need to manually segment the cell membrane. Basically, we are painting the neuron section by section. Depending on the size of the neuron, and tracing a neuron can take a few hours to a few days. And if you need to fully segment the whole neuron, it sometimes can take a few days to even a few weeks. And this is showing a reconstructed skeleton for the 2 ganglion cells. And comparing with the LM images, we can confirm that we successfully identify the labeled cells in the LM image. We are not only trying to reconstruct the cell morphology, we also need to identify the synapses between cells and also matching to the LM images. Here is a video clip showing that how I identify all the synapses onto the ganglion cells. And by manually matching the LM and EM images, I could confirm whether a synaptic input onto the ganglion cells, shown here in green again, comes from the type 6 bipolar cell, shown here in magenta. I usually will use some features on the dendritic arbors as a local landmark to match LM and EM images. And then I will continue tracing the skeleton of the ganglion cell dendrite while looking for synaptic input sites. For example, here I found the ribbon synapse onto the ganglion cell, and I marked the synaptic location and paint the pre- and post-synaptic partners. I went through the LM images to follow the dendrite and saw a matching fluorescent signal in magenta. In this way, I could confirm this presynaptic terminal is part of a type 6 bipolar cell. After reconstructed all the relevant circuits, we then export the 3D reconstructions to Amira to prepare the 3D visualization or videos for publication or presentation. And in summary, as you can see, the whole analysis pipeline is extremely labor intensive. And we also need to navigate multiple tools and the software environment in order to finish the whole project. And because of this, currently only limited size of neuron circuits can be analyzed. We hope with the help of machine learning and deeper learning tools, we could speed up analyzing the EM data and make more novel discoveries. With that, I'd like to thank my PI Rachel Wong and all the members in the lab and our funding agency. And thank you all for your attention.
All right, thank you, everybody. We're going to move along to the demo portion of the webinar now. Hoyin Lai, my coworker, will be leading that discussion here. And just to recap some of the challenges that Wan-Qing just recently mentioned is part of the reason why we started the IGA project with them. This is to help bring together some of the expertise that is in the AVIA software team, specifically machine learning and deep learning and artificial intelligence to speed up and make a lot of these steps that Wan-Qing is talking about a little bit easier and a little bit quicker. So with that, I'll pass it along to Hoyin to start the demo.
All right, thank you, Quinn. Hello, everybody. My name is Hoyin, and I'm the Content Marketing Manager and Senior Application Specialist here at Leica AVIA. So today, I'm going to show you a little quick demo on how to use AVIA to create the type of synapse that Wan-Qing described in the presentation. So I'm going to share my screen. All right, can everybody see my screen okay?
Yeah. Looks good, Hoyin.
Yeah. All right, great. So what we have here is actually a full reconstruction of a couple of cells already. So I will, at the end of this, I can just show right now. So this is basically what the current image looks like. It looks a little flat because we have it's not calibrated correctly. But let's change the calibration here real quick. Back, and you can see there we have this we have this cell traced down through the about 1,300 stacks, and then next to it we have the boutons that are interacting with the two individual cells that are next to each other. So, and we did all of this within AVIA using a tool we call the Mesh Contour Editor. And so to demonstrate how to use it, I'm going to first turn back on the channel. And going back right to the image. So when we were looking at the presentation earlier, we see there is a specific type of feature called the ribbon synapse that forms these very kind of skinny kind of structure that is surrounded by a bunch of little dark circles. So I've already identified one that is on the image, but as we can move along a couple of slices, we can see how it changes over time. And so with that information, what I can do is I can probably start tracing one of the cells that are next to it as potentially one of its partners. So I'm going to trace the cell on the left here, and just to zoom out a little bit. So in the Mesh Contour Editors, there are a couple of different tools that we can use for this. Of course, the most important one is the painting tool. And so I'm going to start with a new slice. And it's hard to see. I have a paintbrush that might be a little bit easier if I also turn it to yellow. And just add the cap back just so I can take a look. And so as you can see, I have this yellow dot, and I can use that to use my cursor to trace along the edge of the cell. And this is definitely not perfect here, but I'm just going to kind of quickly just wrap this one up so that I can move on to the next slide. So when I finish with the drawing, Avia is going to automatically fill in the contour, and then on top of that, it will also move to the next slice. And so so let's go back to the previous slice for just a second. So this will take a little bit of time for while Avia is finding the slices. So so it will generate a prediction based on what I have drawn on the image, and I can use the eraser tool to quickly edit some of these extraneous features out. And now I can also use the paintbrush to enhance the drawing on this, and it will improve the prediction on the other slices as well. So. And in addition to these tools, we also have a fill tool that will allow you to fill in the gaps in the detection as well. So when I'm done, I can move to the next slice, either going up or down. I think I should go down. And then this will take a little a brief moment for Avia to calculate, make an updated prediction. So now this prediction is actually pretty good to what I have drawn previously. So I can go ahead and continue to edit this. And then maybe if I move down a little bit, I can also see that maybe like this part is also connected to my image as well. But I'm actually going to turn off the clipping cap so I can actually see whether or not this is connected. So it looks like this part is connected. So I'm going to turn this one back on and erase that. So there's a couple of different options that we have on the auto drawing. So it uses we can use a UNet model to generate the specific detection. And by default, we have a machine learning algorithm in the mesh contour editor for determining what the next slice is going to look like. But for now, I'm going to turn this off so that I can just keep drawing on the image, just so that we can get a couple of quick slices and you don't have to sit here and watch me wait all day to get the prediction. So. Okay, so once that one's automatically filled out, so now I can continue on with the next slices. And then if I did find out that if my final result was not didn't get the drawing correct all the way, I can always come back later and try to fix it. Okay, just gonna draw maybe like one or two more slices, and then we can wrap this up, and just so we can do a quick demo, and then we can show the result that we've generated. So here the cells looks like it's starting to branch a little bit, so I'm actually going to turn off the stepping mechanism so that I can actually fill in these slopes correctly without introducing too much errors into this. use the flood fill to fill this part. Okay, so let's wrap this up on this slice here. So after I finish drawing the outlines, I can move up and down the image stack to review and confirm what I'm looking at, and then if I need to make any adjustments, I can do that. So for example, here, I will go ahead and extend this down a little bit. And here make some corrections as well. All right, I'm reasonably happy with the result. And when this is done, I can output to the existing mesh that I already have. And there's a couple of options here as well. So I can turn off the smoothing on the to make the final result look looking more natural. But that will be at the expense of losing some of the voxel accuracy. And then finally, when I'm done, and I can also split the object, excuse me, if I'm drawing, starting a new object. And then finally, if I'm just ready, I could just specify which output set. So I'm going to output to the drawn meshes, which is where all of my cells are, and then click complete mesh. So this will generate the surface real quick. And then so you can see now this the yellow outline has turned blue. And to kind of see the full cell, I'm going to turn off the channel. And I'm also going to turn off the orthoplane on the mesh as well. And since this is a little bit dark on the bottom side, so I'm going to uncheck this shade back. So now we can look at the whole data set. Hi. The orthoplane. So now we can see here we have the two individual cells that we have traced previously. And then we have the new cell that I just traced, and then they're all interacting on this one big lobe on the big green cell that we were looking at. So we can turn and rotate this a little bit so we can look at see the interaction. So as we're doing this, we can of course we can further improve the detection. And so this is a very short kind of demonstration of how the tool looks like as we're working to try to get to trace an object on using Avia. So yeah, and also I can just hide one of these cells and see, so I can take a look at just the cells that are kind of interacting with the big object. And also change and in their object set settings, I can also change the transparency, so I can kind of give kind of a nicer look into like how the cells are actually interacting in this case without having them obscuring each other. Yeah. So one last thing is that you'll notice that that actually you can see the boundary of my window, so it looks like a web browser. And in fact, that it is, I'm actually connected to a cloud instance of Avia where I have my data uploaded in the onto Google Cloud Storage, and I can use Avia on a virtual machine to do the trace, to look at the image and to the tracing. And when done, I can also pass the virtual machine to my colleagues or my coworkers or Wan-Qing and Rachel, and have them take a look at the result and see how well I did. So this is a very convenient way for collaboration and sharing, and also a great way to get around hardware limitations. So for example, since Avia requires you to have a GPU to run for 3D rendering and 3D processing, you may not, in this case, you may not have the hardware that can do it, that can do it beautifully. But leveraging the cloud machines, I can actually use a much better processor compared to the computer that I have right now, which is a very small laptop that doesn't have an integrated graphics card. So this gives me like a really nice way to work on the data, visualize the data, and work on the neuron tracing without any kind of hardware constraints on my end. So yeah, so that's part of my my demonstration. So